Hi everyone, my name is Anna Ragney, and today I will be presenting on the ontogeny of shape and integration in the hands and feet of catarrhine primates. Human fossil ancestors all have some part of their skeleton associated with bipedalism, making it a diagnostic trait for the hominin lineage. But they also regularly have skeletal traits indicative of arboreality, leading to contested reconstructions of what a taxon's true behavior may have been. With recent fossil discoveries of juvenile material, an area that should offer some resolution to our understanding of taxonomic differences and the evolution of this trait is exploring how hominins develop their locomotion and what impact that may have had on the adult fossil skeleton. Even closely related primates grow up very differently, so there's potential to use locomotor ontogeny or locomotor development as a way to better understand adult fossil skeletons, create more comprehensive reconstructions, and ultimately understand the evolution of this hallmark trait. Our closest relatives, chimps, for example, spend the majority of their locomotor activity as juveniles engaged in suspension and climbing, but later transition to primarily knuckle-walking behavior. In macaca, we see a decline in arboreality and a large increase in terrestrial behavior through ontogeny. And gorillas and orangutans, on the other hand, show a much less drastic change in their locomotor activity. Now, the external skeleton has been shown to reflect some of these locomotor shifts, including finger curvature. So in this case, a high degree of curvature correlates with a high degree of arboreality, which may shift through development. And in an analysis of the shape of the distal femur through ontogeny by Tallman in 2016, Homo and gorilla showed significant shape change through ontogeny, represented by the strong slopes in this graph, while Pan showed a lack of shape change through ontogeny. In this presentation, I expand this idea out. I'm interested in the hands and feet, which interact directly with the substrate during locomotion. So looking at the hands and feet of 5 primate taxa, I'm exploring how does bone shape change through ontogeny in these primate groups that have distinct developmental trajectories and locomotion. My first hypothesis is that taxa with substantial locomotor changes through ontogeny will show significantly greater degrees of shape change through ontogeny than those taxa with little locomotor change. I'm testing this hypothesis in homologous elements between the hand and foot, focusing on the center ray. And the taxonomic sample includes Macaca mulatta, Pan troglodytes, Gorilla, Pongo pygmaeus, and Homo sapiens. The taxa that experience locomotor change here are Macaca and Pan, so I predict that these taxa will show a greater degree of shape change in their locomotor skeleton than Gorilla and Pongo. Now, because human behavior is so diverse, the prediction for Homo is left open. I'm also testing a second hypothesis. I hypothesize that taxa with coupled function of hands and feet will have significantly integrated chiridia. Now, this one I'm going to break down a bit. By coupled function, I'm referring to the functional roles of the hands and feet. Now, for all the non-human taxa here, they use both their hands and feet for locomotion and are therefore functionally coupled. Humans are not. Integrated refers to morphological integration. This is an assessment of the degree by which traits covary or correlate with one another, either functionally or developmentally, like these bivalves in terms of valve thickness. Now, chiridia is just another word for hands and feet. So with this hypothesis, I predict that macaca, pan, gorilla, and pongo will show significantly greater correlation between hand and foot shape through ontogeny than Homo. This matters because while the human foot is part of a suite of traits allowing for bipedality, the manipulative abilities of the human hand are also unique. So how did the hand and foot become decoupled in humans? There are several hypothesized mechanisms. Darwin suggested that separate selection factors have impacted the hand and foot independently, initiating the functional decoupling. Rolian et al. In 2010 hypothesized that selection in the foot resulted in features in the hand through developmental covariance, which were then exapted for manual dexterity. Finally, Almethia et al. 2015 hypothesized that the human hand represents the ancestral hominoid condition while the foot is derived. Because the two circled hypotheses here do not rely on changes in one region of the body impacting the other, they would be associated with low integration between the hand and foot for Homo alone, matching my prediction. If my prediction is not supported, and instead it turns out that Homo has integration levels similar to the other functionally coupled taxa, then the hypothesis of Rolian et al. 2010 is supported. So with that all in mind, I'm focusing on two elements within the hand: the capitate and the third metacarpal. These bones are functionally relevant for load-bearing in the knuckle-walking chimps and gorillas, and they also represent some of the earliest bones to ossify within the hand, which allows for easier identification in juveniles. In the foot, I targeted homologous elements, the lateral cuneiform, and the third metatarsal. These bones are also among the first to ossify and are particularly relevant in comprising part of the human arch. The study sample consisted of 21 individuals per taxon categorized by dental development, and I collected shape data from micro-CT scans using the following micro-CT settings. In order to test these hypotheses, I had to quantify the shape of the bones, so I did this by extracting just the bone's surface from the micro-CT scans in Volume Graphics, Studio Max, and GeoMagic. I then isolated several 3D landmarks to capture the overall shape of the bones. These landmarks are present on both the youngest and the oldest individuals and across taxa, and I then aligned the landmarks to test the first hypothesis. To test hypothesis 1, we need to quantify the correlation between shape change and size change. So for each bone, I calculated the difference between each taxon's average juvenile shape and each individual. It's quantified as the log Procrestes distance. Then for each bone, I calculated the difference between each taxon's average juvenile size and each individual. It's represented by the log centroid size. To test change, we're plotting size change and shape change and performing linear regressions in each bone. If there's no change in shape through ontogeny, the slope will be 0, like on the left. If there's a significant degree of shape change, the slope will be different from 0, like on the right. On to the results. Here we have that regression in the capitate. The x-axis is a size proxy, so the left side also represents juveniles and the right side represents adults, and the y-axis represents the degree of shape change. All taxa show a slope significantly different from 0 here except for Homo. In pairwise comparisons, macaca shows a significantly greater degree of shape change than all the other taxa. For the third metacarpal, again, all taxa show slopes significantly different from 0 except for Homo, and in pairwise comparisons, Pongo in blue is significantly different from Pan, Homo, and Gorilla, and Macaca in magenta is significantly different from Homo. In the lateral cuneiform, again, all taxa show slopes significantly different from 0 except for Homo, and in pairwise comparisons, Macaca shows a greater degree of shape change than all the other taxa. Gorilla also shows a greater degree of shape change than Homo. In the third metatarsal, as in all the previous bones, all taxa show a slope significantly different from 0 except for Homo. In this bone, Homo stands out as being significantly different from Macaca, Pan, and Gorilla. So what stands out here in sum? Firstly, my hypothesis was not supported. In no analysis do Pan and Macaca alone show significantly greater degrees of shape change through ontogeny than the other taxa. What also stands out here is that all taxa except Homo show significant shape changes through ontogeny. And while that may lead you to believe that Homo is distinct, in all bones except the third metatarsal, pairwise comparisons show that the degree of change for Homo is only significantly different from another taxon here and there. So, though it's the only taxon with insignificant shape change through ontogeny, Homo is not actually statistically different from the degree of change we see in other taxa except in the third metatarsal. What this tells me is that behavioral locomotor shifts do not correlate significantly with shape change any more than other factors through ontogeny. Hypothesis 2 predicted that all taxa will have significantly greater integration levels between the hands and feet than Homo. In order to test this hypothesis, I compared integration levels across the samples using the geomorph package in R. I did 3 analyses: adults only, juveniles only, and the full developmental sample, and this was to isolate functional versus developmental similarities or differences. So looking at adults only, I found equivalent levels of integration between the capitate and lateral cuneiform as well as between the third metacarpal and third metatarsal. Looking at juveniles only one significant difference showed up. Juvenile pongo showed significantly greater integration between the capitate and lateral cuneiform than macaca. These results indicate that for the most part, the hands and feet are similarly integrated in adults and juveniles, regardless of functional locomotor similarity. When I looked at the full developmental sample with juveniles and adults combined with adolescents, macaca showed significantly greater integration between the capitate and lateral cuneiform than pongo and homo, and significantly greater integration between the third metacarpal and third metatarsal than homo. So contrary to predictions, homo only showed distinction from macaca and only in the developmental sample. The surprising amount of similarity in integration levels across the taxa points to a conserved pattern of hand and foot development and integration, especially among the great apes. In conclusion, the major takeaways from this project are that hand and foot bones significantly change shape through ontogeny, except in Homo, though statistically Homo's lack of shape change rarely constitutes significant difference from the other taxa. Behavioral locomotor shifts do not correlate with shape change any more than other factors, and integration between the Homo hand and foot is similar to other primates. Now, these results are in contrast to some previous work. Tallman in 2016 analyzed shape change in the distal femur through ontogeny and found that humans and gorillas show significant shape change while Pan did not, whereas I found that Homo showed no shape change. In 2012, Green and Alemseged analyzed shape changes in the shoulder through ontogeny and found humans to be the only taxon to change shape significantly through ontogeny. They also found that significant changes in shape correlated with shifts in behavior. Those trends were not seen here in the hand or foot. What this means is that shape change through ontogeny may be region-dependent and not a systemic trend throughout the skeleton. Green's study also went further. They reconstructed a juvenile Australopithecus afarensis as having ape-like development because there was no significant change in shape through ontogeny. Now, that study was performed on a different bone of the body and used linear measurements, and again, perhaps different patterns of shape change occur throughout the body. However, if I were presented with the same fossil hominin results in the context of my data set, I would come to the opposite conclusion of Green and Alemseged. In the context of my data, the fact that this juvenile fossil shows no shape change would make it human-like in its development instead. Now, that being said, I also found that humans' lack of shape change was not statistically different from the degree of change seen in other taxa, so it would be difficult to draw conclusions. Now, what do our integration results mean in the context of our evolutionary hypotheses of hand and foot decoupling in humans? Because Homo shows a largely similar pattern of integration between the hand and foot despite a functional decoupling, the hypotheses that favor distinct selection pressures between the hand and foot, which would result in low integration values, are not supported. Instead, the hypothesis supported here is that of Rolian et al. 2010, which posits that selection acted upon the foot resulted in similar anatomical changes in the hand, which were then exapted for manipulative abilities. This hypothesis particularly relies on there being high integration levels between the human hand and foot. The hands and feet of Homo showed just as much covariance as other primates, supporting the idea that the dexterity of the hand is an exaptation from anatomy reflective of adaptations in the foot. Now, the brief caveat here is that I'm only focusing on 2 bones of the hand and foot, and if I focused on the fingers and toes or included more bones, this conclusion may be different. Other areas of future work include analyzing juvenile fossil hominin hands and feet for comparison to modern primates. It's also unclear how bony change through ontogeny differs throughout the skeleton across taxa, which may be why different research teams have found bone-specific patterns in primate comparisons. So I plan on approaching this with a much broader mammalian sample to explore that systemic versus regional dichotomy. I look forward to this future work and thank all of you for listening. I particularly want to thank the following institutions, individuals, and funding sources. Thank you.